Hello, welcome to another Talk Wildlife, and today I am absolutely delighted to have with me a person who has written a second book, which I have to say was my most anticipated book of the year, probably my favourite read so far of the year, and that is Erica McAlister from the Natural History Museum. Hi Erica, how are you?
I'm very good.
I promise.
Yeah, I'm wearing a mask, so I hope I don't sound too muffled. But
right.
I have to be careful.
You don't now, but also tell us what you're doing. Right. First of all, tell us what your title is at the Natural History Museum.
My title that you know now. So I'm a senior curator.
Away now. I was going to act all clever then, but you're doing it away. But go on. What's your title?
Okay, I'm senior curator of Diptera and Siphonapptera.
And for those of you who don't know, that's fleas.
Flies and fleas. So yeah, I do all the jumpy, bitey, fly, stabby things, which is like the best things on the planet.
So just a quick question on that title then. So flies and fleas, which came first in your sort of, was it like, I'm going to study flies or I'm going to study fleas?
Oh no, I got given fleas, as it were. The flea person retired, so I inherited the fleas, which have to say, we in the museum, most of the time, we incorporate collections into the main collection. So I have a big collection behind me, which is all these flies, beautiful flies that were collected by this guy during his lifetime. And I will database them and put them in the collection. But the flea collection came from a rather more famous person. And the majority of our flea collection came from Miriam Rothschild.
Really?
Yeah, and it is in these mahogany cabinets, and they're like velvet lined drawers. And it is like, oh, you walk in, and I don't stroke many cabinets. Well, I do stroke quite a lot of cabinets, which is one of my weirdness. But when you go and see the flea collection, it's just like, oh. And then some of them, most of them are organized taxonomically, but some of them are organized in what she was studying. And so there's one that's my favorite, and it's called Monstrosities. And it's just like mutated fleas that she's found, and she's like slide mounted and done everything else. And then there's the genitalia cabinet, which, you know, every woman needs a genitalia cabinet, and all of these sorts of things. It's brilliant. It's beautiful.
Right. So before we get on to flies, I just want a quick question about fleas. I know that there is more than one species, even in this country, but how many species of flea are there in the world-ish?
Oh, you put me on the spot. I think it's 2,500.
Really?
But I could be lying. In the UK, I think we're at 68.
Right. I wonder if Andy Musgrove's seen all 68.
My favourite, do you know what the largest flea is? Yes.
No.
My favourite is the largest flea is about I don't know, just shy of a centimetre, and it's found on the mountain beaver, which is not a beaver, and the largest one in the UK, which is hilarious, because it is well over half a centimetre.
That was Andy Musgrove probably telling me that he has. Oh, well, that's quite big.
It's very big, isn't it? See, there's my finger for a scale. So what we say in that is just shy of a centimetre, maybe I don't know, 8 millimeters. But that's found on the mole. On
a mole?
Yes.
I can feel another interview coming on here. So let's come back to flies. But first of all, I have to say, because you've told me what you're actually doing at the moment and what you'll be doing while I'm talking to you. And I thought, now I'll let you break that to the world. So
yeah,
everybody knows you pin your fly and all that, but what are you actually doing at the moment?
So in the book, there's loads of beautiful images, yes, and they were wonderful. And what is very handy is that a lot of those specimens come from the collection here. So I brought them out of the collection, but we're on a massive digitization program at the moment. We're trying to barcode everything. So right now I'm sticking tiny little barcodes on flies. So these flies have got to go, they were imaged for the book, and I've taken a photo of their labels, a professional person, thankfully not me, has taken a photo of those beautiful images. And now I add the little barcode and back they go into their home. But what's cool is that these now will go online as well. So you'll be able to see all these wonderful images as well. So yeah, so it's opening up the collection for everyone. So I mean, at my present rate, I'm going to take personally about 200 years to complete it. So luckily we have digitization programs to speed up this process. So everyone around the world can access all this material, which is a way of, we're repatriating all the data. We're giving back finally. So we're looking after it, but everything, the images, the data is going back to everyone. But I'm getting sidetracked because I just keep looking at flies go, cool, look at that one. And
it's brilliant, but I bet there's not a person watching this interview. And hopefully there is one, but I bet there's not a person that will watch this interview that can say that their day was anywhere near as interesting as what you're doing.
Yes, barcoding flies.
Barcoding flies. Today, I will be mostly barcoding flies.
It's what I do a lot.
Speaking of so this is said book. Highly recommended. Christmas coming up. Definitely should purchase. If you haven't got the first one, get that one as well. Awesome book.
Do you know what? What is amazing, what I love, and I don't know if it's going to happen with the next edition, if you take the cover off.
Oh, I know.
Isn't it beautiful?
It is. But then, you don't want to take the cover off, but at the same time you do. So
I know.
Too.
But when it was our publisher, the guy who's been doing so much towards this, he said, Erica, what do you think of this? And I'm like, oh, that is absolutely gorgeous. I just love it.
The whole thing, I have to say, I've got the paperback version of the other one, but I'm now taking the dust cover off. But it's not just the cover, right? It's not just, but look at this.
I know.
It meant we could actually just spend the next hour just talking about the virtues of how the book's put together, because it's brilliant. And there's a fantastic picture in here. I don't want to get distracted, but if it comes easy, near the front, I'm sure it is. Some of the photography is absolutely outstanding, but there is, I'm sure it's, for a start, this is not actually my favourite, but look at that.
I know. This is the thing. Somebody was pointing out, and I have to be careful because the butterfly curator is in today, and she may hear me say this. So, everyone goes on about how beautiful they are, and they just put up a picture of a butterfly, and I was like, it's nice. It's got a pretty pattern, but can we just check this out for a sec? Because these are like robust, chunky little nuggets of wonder. And what with the eyes, the legs, the genitalia, I mean, just come on. They're just superb.
And before I ask you the first question, I'm going to show another picture because that wasn't my favourite in the book. This is my favourite picture in the book.
Stomachses. Yeah, it's beautiful. That mouth part. Can you imagine that piercing you?
Do you know what? I'd almost let it, just because it looks so amazing.
I do this with mosquitoes. I'm like, wow, look what's going on. Can you imagine it's moving under the skin and my partner's like, oh, well, leave her.
You must be a joy to live with.
Don't touch that fly. Oh, can we move it outside? Not quite yet. I haven't identified it. Oh, okay, fine. Are you listening?
Mind you, having said that, I'm not quite as sort of focused as you, and I don't go barcoding flies, but I am the same. If there's something in the house or in the garden, or I've got to identify it, and I don't pretend to be sort of a specialist in anything. You've got to do it. You can't let something fly through your house and not know what it is.
And the book starts with sitting in my garden in an August morning, and I noticed the flies. So I was sitting with him, and he said, "Why are you so amazed?" And I said, "Look," and I picked up one of these flies that had just emerged from its pupal case, and it was doing the whole head deflating, inflating. And I said, "Watch this," and he's like, "Whoa, what's going on there?" And I said, "I know. It has just blown its head up to emerge from the pupal stage." And he goes, "But it's grey." I said, "You will wait." And he goes, "Well, how do you know it is?" And I said, "Just trust me on this." And he watched the exoskeleton harden. He watched the eyes collapse. He watched the wings unfold. And afterwards, he just went, oh, and he sat there for 20 minutes with a fly on his finger, just going, this is amazing. This is amazing. And it was just like, at that point, what we forget, we genuinely forget how much nature is around us, and how that process, which is arguably one of the most awesome processes in nature, and it's there, it's in our garden, you all can see it. And his little mind, he's a male, his little mind was blown by, this. And he was like, okay, I'll give it to you. There's something quite funky going on here. And it's like, yep.
Oh, yeah, absolutely. Well, we're going to start, actually, because your book's called The Inside Out of Flies. I'm going to start on the outside. Because I want to talk to you first of all, and you in the beginning of the book, before you get to the first chapter, you talk a bit about this anyway, so it's still sort of in the order of your book. Exoskeletons. So everybody knows insects have exoskeletons. But, these are quite, you said, I think you said the word armoured earlier on. I mean, it's a really special development, an exoskeleton. I mean, it's not just some soft outer material. Tell us what its makeup is. How is an exoskeleton made up? What's it made of?
Well, so sometimes it isn't armored. That's the whole thing about a maggot. But so arthropods, all arthropods have this exoskeleton, and most of we think about crabs and spiders and beetles. They have a really toughened exoskeleton. Lots of chitin in it. Like one of the beetles, you have to drill through it. That's how thickened its exoskeleton is. These are hardcore. And these are the skeleton on the outside. This is an amazing structure that enables them to do all sorts of stuff that we just can't do, because they have a defense mechanism, they have all sorts. But it has to be flexible as well, because imagine walking around with an armor all day, a suit of armor, but you've got to have ridges and things like that. So they have weaknesses and whatever, and they have thin exoskeletons that they've made into like wings and things like that. So there's a huge amount of variability within this exoskeleton. Plus, when, for instance, the larval stage has a really, really thin exoskeleton, because they need to just be floppy and maggot-like and get everywhere. So the fact that it changes over time, it changes where it's on the body, it's changed it into different forms, is a unique and arguably one of the most important things that have enabled this group of animals to just conquer the planet.
I have to say, yeah. I can't disagree. So so exoskeleton, we know it's hard, all the rest of it. You mentioned maggots. Yeah. So floppy. Maggots, if anybody's ever held a maggot, which I'm sure everybody has, they're sort of soft and squidgy. They're not hard. So what takes place during metamorphosis for it to be able to build an exoskeleton?
Well, the exoskeleton is there all the time. It's whether it's scleroterized, whether it has got this extra chitin layer in, where it's thickened. And so what goes on during this pupal stage is actually the most funkiest thing on the entire planet, because it's not a complete reorganization of body tissue, but there's a major reorganization. And we've been studying this fly. We've been studying the Drosophila for over 100 years now to properly look at what's going on. But it started way before that, like Jan Swaneman started it 300 years ago, looking at what was going on. And we're realizing that there's some distal cells, there's internal cells signifying, that are giving signals telling the different body parts what to form.
So it's also... You talk about the Hox genes, are you?
Yeah, and things like that. So they're telling everything what's going on. So they've basically, the larval form didn't need anything. The larval form needs a gut, and that's about it. It's absolutely, it has to just eat for its life. I'm so coming back as a maggot, I tell you. I am, thanks to lockdown, I am so practiced at this life eating thing. And then it is like, right, then we're going to completely alter the body form. So that what they're doing, and if you look during the pupal stage development, and now thanks to technology like micro CT scanning, we can look at what's going on within the pupal case while it's developing. So we can see all of these things happening, and the development of limbs. And this is quite an important thing, because the adult stage is the sensory stage, and it is the dispersal and reproductive stage. So before that, there was no genitalia. So this is all coming about during the pupal stage, which is quite fun. But obviously the pupal stage is quite small. It's like, so it's not like they can stretch out, you know. I'm not one that's had my own maggot, but I've been told about all the stretching and kicking that a baby does, but they can't do that. And so when they emerge, they will then have to stretch out, and it's this when the sclerotization happens. So this is when it takes it from a very, very soft, vulnerable stage to the hardened, hardcore insects with their wings and everything, that stage.
Right, so I have to ask you, how? Where does that come from? Is it something that it'll have eaten during the maggot stage that will form that, or where does that come from?
What do you mean, where does what come from?
Well, you know, you said it hardens.
Yeah.
But
so this is a neurological signal that you're outside now, and go for it. So then you get a physiological response where you start getting this chitinization process going on. So there's a hardening process. Of course,
it basically, it's almost like cell generation.
Cell hardening, maybe.
Cell hardening.
So it's a chemical response going on that's going, right, we're going to chitinize.
Brilliant. And now I get it. I'm on it.
Yeah.
Anybody ever asks? So I'm going to share screen because I've managed to download a couple of pictures, and I think it will be quite useful to do that, if I can remember which screen I want to share. Yeah. So we're going to share this one. Right, okay, the reason I want to share this one is because there's a couple of things I want to talk about on the outside that are going to sort of then link us to the inside. So first of all, let's talk about the anterior spiracle. So, and in fact, no, let's just stop a second, because you said something in the last interview, and if you haven't watched the last interview, you should, because it's highly entertaining. So you said something in the last interview that I didn't pick up on, and you mentioned it in the book, and it's something that you never really think about, and that's that we all know flies can see, we know they can smell, but you mentioned that flies can hear. Now, I'm not saying that I did know that, I'm not saying I didn't know that, it didn't even cross my mind that flies could hear. But
how do you think a mosquito?
Yeah, you mentioned the mosquito could hear. So let's talk about that first, because I found that, I thought,
right,
yeah, I've never thought about that before. Tell us about how flies hear.
So you see this bit on the head and the antennae. That antennae is the organ, the sensory organ for hearing. And what it does, it converts mechanical waves into auditory responses, so it can hear the vibrations. So when a male mosquito is sitting there waiting for his lady love, and she flutters past, he listens to that vibration of the wings. And in very much, I describe it in the book, it's the cocktail party. So, you're sitting in a room and somebody's talking about you and you just hear your name. It doesn't matter how many other people are in the room, you can hear it. And what he's doing is he's listening specifically for his favorite woman. And she has to be fecund, of a certain age, of a certain species, obviously, and sex. And he's listening for that. He can hear that all glowingly. And we've done this with Drosophila as well. Clever people have done this with Drosophila. And they're realizing that flies are really good at aging other flies as well. So they know when a fly is ready. This has been all sorts of, there's a sex lab, I'm sure it's got a proper name, in Singapore, and they're looking at sepsids, and they do all sorts of understanding how they do this. Now, the antennae, they've got a Arista on it, some of them, this one has. And in the base of the antennae is the Johnston's organ. And this is the bit that properly enables them to hear. And it was described from Johnson, I can't remember what it was now, 100 years ago, in mosquitoes. And mosquitoes have, some of them are the same level of hearing as us. They are so good because of the amplification. They've got this one here on the Arista, you can see a load of flagella, loads of like spikes coming off it. And they, you know, you're increasing the surface area, so they are so much more sensitive to different vibrations. So they're really, really good. And a mosquito will definitely listen to you first before she's close enough to smell you, or get mechanical cues and things like that. So they're very important. But I presume what you're going to talk about is not the hearing on the head. You're going to be hearing on the thorax.
Well, we're going to come to that, but hold that thought, because this is probably going to be a really, really stupid question. And you're probably going to look at me and go, yeah, how am I supposed to answer that? But, okay, the hearing vibrations. So what do they hear generally? So if a fly is buzzing against your television, which
you hear vibrations.
Yeah.
You're hearing the vibrations on your eardrum. So those different vibrations are being interpreted by your brain into different noises. So exactly the same thing. So think about your tympanic membrane. Think about that, and think about all of these different ways. They have just a different mechanism for picking it up. So they're hearing as well. I mean, I don't know how they, you know, because I can't see into your brain. I don't exactly know how your brain works.
No, do I.
You know, and your poor wife. But that's the, again, it's exactly, they're just picking up this, and they're translating all of these mechanical stimuli.
Right. Okay.
Oh, you got that?
Yeah, there's another interview in that somewhere. It's a massive subject I want to talk about, but I'm conscious that I've got other bits and pieces I want to talk about, so maybe we'll come back to that another day. So talk about the thorax then, because you mentioned the thorax. You know, the smell through the feet. So
taste on the wings, smell through the feet, like you name it, every bit of their body is just like it's a sensory overload. It's fabulous. But they can also hear. Some of them can hear on their thorax. And the thorax is the central segment of an insect. Insecta is the three tagma, so head, thorax, abdomen. And theoretically, all insects have this segmentation. Some flies have done some crazy bits, but we might get onto that later. Now, the thorax is predominantly where you've got the locomotory structures associated with it. So the wings, the legs, etc. And the head is where all the sensory structures are meant to be, you know, the hearing, the sight, the taste, etc. But flies don't just stick to that body plan. And there's a group of flies in the family Tachinidae, and they're the parasite flies. Or basically, I call them the spiky chunk flies, because they are the spiky little ones. They include the hedgehog flies and things like that. They're adorable little chunks, most of them. And hence, the common name is the parasite flies. I think you could tell what their life cycle is.
Yep.
They are parasites and parasitoids, and so the females have to go and find a host for her offspring. And women are very, very dedicated to ensuring their little maggots have the best life. Now, there's one Ocmea, which is this beautiful orange little tachinid, whose larvae live in that host of grasshoppers. And grasshoppers are quite fast, and they can boing away quite quickly. So she's got to use all sorts of craziness. And she's tiny. She's a tiny little fly, and a grasshopper's huge. And so she has to develop more mechanisms to be able to ensure that she gets her host. Now, our ears are separated by this big gap in the middle. Most of us have brains. Some of us don't. But we're able to triangulate noise quite well because of this large gap. Now, they've got no real gap, so they can't triangulate in the same way. So what she's done, above her anterior spiracle, just on her neck, basically, just lower her neck on her shoulders. She's developed this organ, this hearing organ, which amplifies all incoming noises and amplifies the left and the right side. And so she's got a much louder, she's able to triangulate better. Really tiny noises. It's this resonance thing, isn't it? It works in like this cradle system that amplifies this noise higher. And what is so funky is that it really does work well, extremely well. In fact, we are mimicking it now to make speakers, really tiny speakers. And what's really cool is hearing aids, absolutely minute hearing aids based on this parasitic fly, which I think this is one of the things I really enjoyed about this book, was reading all these secondary effects of all this that we take for granted, so many little things, but nature's done a really good job of doing lots of things before us. And this is one of the reasons I'm so concerned about looking after nature, because it's got so much to tell us. And this is why it's quite fun. But I just like the idea of this tiny little fly. And I don't even know who first looked at it and went, you know, that's a brilliant idea. It's fabulous. Love it. It's like, oh.
Yeah. It's crazy, really. I mean, when you read the book, it's like, you know, there's sort of fact after fact after fact that is just fascinating. Moving on from hearing, because you've answered my question. The problem is, in answering the question, you opened up a whole new doorway in my brain to the evolution of flies that you know, we'll have to talk about sometime, because no, let's ask the question now. So, you might not know this, but you probably will. But the first common relative between us and flies was, what was it? What kind of thing was it?
Common relative between us and flies?
Yeah, first common relative. I mean, it must have been. So where did flies? So obviously, you look at the evolutionary tree. If you go back far enough, there is a common relative. So we'd have had a common relative, but at some stage they'd have split off. So what did the first? Let's ask the question of what did the first ever relative of the fly look like?
Well, the flies are in the Antlinomorpha, and that group includes the scorpion flies, the fleas, and the flies. And then you've got to go back to basal insects. And insects are a crustacea, so you go back via crustacea. And then in the arthropoda, you've got the Trilobita. Before you get back into and it goes back. I'm not an evolutionary biologist.
No, I just, because when you were talking,
I've never really worried about humans. I've never really studied humans.
I'll forget humans. So I know your next book's a kid's book coming out soon. Yeah. That's going to be amazing. I think the book after that needs to be sort of the evolution of flies.
No, I've got a different book. It's already started, but I'm not going to say anything yet, because it is in the early evolutionary process at the moment. I'm in the primordial soup stage of the third and final chapter of flies. So.
Right, I'm looking forward to it already. When's it out?
Oh.
I bet you publishers say that to you all the time.
Yeah, there's a few hassling me. I'm like, okay. So, yeah.
Right, moving on then. Sphericals.
Yes.
Right, so everyone, I'll point it out. So there's an anterior spherical. There's another one down here, posterior spherical.
Anterior and posterior. Now, they've also got other this is the adult fly. Larval flies, as everyone knows from my first book as well, I've got a bit of a thing for anal sphericals. I mean, who doesn't? And, you know, and larval Diptera have got, some of them have basically just 2 sphericals, some of them have loads of sphericals. It really does depend. And the larval stage, they can obviously perspire through their skin, as it were. But the sphericals, let's go with the anterior and posterior with this fly. And what is amazing is what they link to and how it works. So they don't have a lung system. The closest relative to them that does is spiders, with a basic lung called a box lung. And everyone I grew up calling this passive. So it was just passive diffusion of oxygen, but it's not totally passive, because they will, you know, resonate their thorax and things like that and abdomen. So they will kind of pump it around the body. So, you know, we did talk for a long time about this, but that's not actually the case. But they have this amazing tracheal system. So it kind of reminds me of tripe. You know, when you've got all the rings.
Right, okay.
Yeah, no? Shall I get a better analogy?
No, that'll do. I'm sure that there's some people out there that remember what tripe was.
My first job was working in a kennels when I was very little, and I had to cut up the tripe, and it's forever there. It was not pleasant. Anyway, yeah. So you've got this series of ring system, and it's amazing. So it enables oxygen and carbon dioxide to coming through the body, and the flies, as with other insects, are full of pumps. They've got hearts everywhere, which I love. They've got several hearts in their head, hearts in their thorax. They've got some hearts in the leg. They've got even a heart in a genitalia, some of them, which, I mean, what more? More can you say about why these flies are so amazing? So they
The
they
heart
have
really does rule the head then.
The
Yes,
heart
really
in
does
this
rule the head.
quite literally. So you need this gaseous exchange to go around, and it is extraordinary. But trying to find a diagram, I think they're doing one now. It's coming out of the American Museum of Natural History, AMNH. They're looking at doing some really good imaging of the tracheal system. It's very complicated, and yet we have, and this again is one of the things I found in writing the book, with very little information. It was like, everywhere I went, very little information. I was like, gosh, so much to learn.
Well, it's interesting because you've sort of mentioned hearts and, you know, tracheas and all the rest of it. And you know, most people in the world at some stage have swatted a fly, and it's been on the wall and it's not gone well, because it's sort of basically just burst all over the wall. So it gives you the impression that what you have here is you have an external skeleton that in effect is just basically a sack full of goo. Now, you've already mentioned them, so I'm going to go on the inside now. This is what that goo looks like. This is a picture I've managed to find on the internet. And reading the book and seeing this picture will, I hope, astound people as to what that goo actually is. Because like you said, you've mentioned, you know, hearts and all the rest of it. So here we have the inside. Now, I want to start at the front. We've got the eye, and again, the eye could be a whole interview on its own, because the eye is incredible. But just behind the eye, that greeny bit, it might seem obvious, but that's the brain, yeah?
Yep.
Right. Okay, so tell us about a fly's brain. Is it...
in comparison to us, it's quite simple. After this year of politics, I disagree. But anyway, this is, so we have now started properly mapping it, which has been amazing. There's been some absolutely outstanding work coming out, you know, in the last couple of years, where they start mapping it. And they're realizing that although it has a lot less neurons than us, I think it's got 100,000. I should have read my book, shouldn't I? You've got multiple connectivity between all the neurons. So it depends which one's firing off to what message is going on. So it's this, just imagine a really complicated highway of things going everywhere going on, this 3-dimensional highway. And it responds quickly, really quickly. So you've got this neurological pathway connecting to the legs, the wings and everything. And it doesn't necessarily have to go to the central processing part of the brain. It can be bypassed and go straight to the legs and wings. Hence why when you do try and swat a fly, it's able to process that information so quickly and act on it. So they will see faster than us, as it were, because they are processing images faster than us. They're reacting everything. So when you're coming at it like that, they're like, oh. So you know those brilliant like slow motion scenes in The Matrix? Basically, that's what flies just go ooh all the time. So you can, next time you do it, put a little sound effect going ooh, as they do that.
I will. I'm going to take the voice recording of you from this. I'm going to play it as I do it. Right.
Yes.
Okay, so the brain. So here is the brain. I think, yeah, here's the brain here. Right. Now, what I've noticed here is that a fly is sort of almost upside down from us, because in our back, you know, we have our nerve cord. The nerve cord, this is the nerve cord here, isn't it? So it's almost running down. If this was the back, it's running down the sort of almost down.
I think this is a little bit more artistic than it is actual, because you wouldn't necessarily, I don't believe it runs along the base, as it were, but it's very complicated because the gut is well, it depends actually on what fly, but the gut will take up quite a bit, and so, but you would get more of a connectivity between these.
It's not overly scientific. It's sort of
no, and it's a bee as well. But anyway.
So number 19, for anybody that's looking at this, number 19, that is the nerve cord, isn't it? Right. Okay. And then we've got the green bits and the darker green bit, and that's the gut.
Gut.
And like
I love the gut. I mean, you've got the fore, mid, and hind gut. Yeah. And the fore gut is in the thorax, and the mid and hind gut are in the abdomen. And so much goes on in the gut. It's amazing. And we're beginning to properly look at these, because this is really important when it comes to disease transmission and things like that. So with the plasmodium, so when that little mosquito has sucked up some plasmodium and gets into the gut, we're trying to actually, that's the bit where we can stop it getting through the gut lining back into the saliva of the mosquito to be passed on to another host. So this is a really important bit we're trying to properly understand. And there's all sorts of things, like some adult flies just don't have guts. They've got enough reserves during the larval stage that they're like, whatever. And I believe it's in the book, so I show the difference between a housefly or a blue bottle and a botfly. And I love a botfly. I mean, who doesn't? They're adorable little fluffy fluffs. And they are, I've got some botflies. Get a botfly out. Do you want me to get a botfly?
I tell you what, wait until I stop sharing, because otherwise it's going to be so small. But when I stop sharing, you've promised us.
I'll show you a botfly. So botflies, basically, the adults are just there for one thing, what adults do best. And so they just need a lot of reserves, and they haven't got time for feeding. They haven't got time. They've got to go make a family. And so that whole bit of the gut is gone, which is amazing. But they've got, they have other bits to make up with it, and a lot of muscles. So the thorax inside will be mostly muscle with a lot of flies, which is quite amazing to see that. And then you've got, what's 20? I presume that's your Malpighian tubules, is it?
Oh, thank you.
Is it?
Number 20, I'll tell you what. Number 20 is the Malpighian. Yes, absolutely. Yeah, you are. Right. I tell you what, you should be sort of head curator at a Natural History Museum.
Thanks, honey. So this is their kidneys. So this again is quite amazing. So they haven't got a blood system like us. The blood system is open. So they are in this, like, whatever. But they've got to filter it, and, you know, exactly the same way as us. They've got all sorts of things. But one of the things that flies have done funky is some of them have used them to make bioluminescence in their kidneys. Love this. Can you imagine? And so they glow, and they use them to attract prey and all sorts of things with their glowing kidneys, their bioluminescent kidneys. How cool is that?
Yeah, again, you know, there's just so much we could go on about. So because you said of an open blood system, that's in your book. So what's this? You can see my arrow here.
Yeah.
What's this here?
What is that?
System. Number 7, I think it is. Yeah, 7. The red one. What's the red one?
Don't know, what is it?
Because the red one, it says it's the dorsal artery.
Dorsal artery. Yeah,
Now,
so they will.
You said it's got an open blood system.
Yes. So, but that's a heart that goes all the way down.
Right.
So yeah, so you've got things going all along. So you've got pumping systems to enable it to do that. But it's got your veins in the wings. So that's where you're pumping blood around again, remember? So you've got to have all these sorts of things going on, blood around your legs, all sorts of things. So it's quite important that you have this fluid system and this regularly changed fluid system. What is amazing about insects, we talk about hemoglobin in humans.
Yeah.
And flies can have quite a few. No, we have one type. Flies can have quite a few. I think chironomids, the non-biting midge, has 13, which is well cool. And they're not like little ones like us. They're some of those massive globins. And they don't just use them to transfer oxygen around the body. They can store oxygen. So they can nicely sit out in an anoxic environment going, well, we've got our own air, thanks. We've just stored oxygen in ourselves, which is something, these hemoglobins is something we are definitely interested in, because can you imagine if we could do something similar?
Yeah.
And where we could travel and all sorts of things going on. One day I like the idea of being a human scuba diver. Just going in with this plastron over me and yes, the sea floor.
Oh, right. Yeah. You see, now you're taking me on a tangent again. But we've got to mention it. The saline fly, is it?
Scuba diving fly.
Yeah, that scuba dives in, because I'm actually going there is it Mono Lakes in California?
California.
And I had in my mind my list of birds. There's also a fly on it. I've got to go and find that. I've got to go and find it. Tell us about that. How does that work?
I also, when I went to Los Angeles, the tar pits, and I saw the little tar pit fly. Honestly, it's like, I should write a list of flies I want to see around the world, like you crazy bird people, because it was just like, oh God, everyone's like, why are you so excited? Look at the fly, everyone. Like, no. So this is it's this fly in California, and it's amazing. It's a ovipositors. They're called shore flies or brine flies, and so you can see where they normally
brine flies. That's it, brine flies.
Yeah, but these are like way more funky than the average animal, because the mono lakes in California are known for what? They're super saline.
Yeah.
Like they are the most saline aquatic environments in the world, because it's not like the sea. They're fed from these all these tributaries, and there's nowhere for it to go. So it just evaporates, and so the water gets more and more and more concentrated. So basically few animals live there, because they're just like, oh God, no way, this is awful. The fly has looked at this environment and gone, nope, I'm up for a challenge. It's like flies.
For me.
Yes, flies are like, can I think of a more extreme environment? Hold on, strap on, I'm going. And they are like the best explorers because of this. And so they physicists, I love this. I love getting physicists involved with biology stuff, because they like, we just go, that's amazing. They go, now hold on a minute, what is going on? So they've measured the angle at which this female penetrates the water, and it's a specific angle. And she has to, which forms a bubble around her, a plastron. Now, lots of insects can do this. So we get them in the UK, water beetles and flies and all sorts of other creatures can have this plastron. But thanks to the chemistry of I was about to say ours, of the exoskeleton, these other insects could not cope. But the hairs and the hydrocarbons that this little fly is producing is enabling her to penetrate these really saline environments. What is so funky is that her eyes are exposed. She doesn't cover the eyes, which, how does that happen? How on earth does that happen? The magic of it. And so she climbs down, she scuba dives down to the base, and she lays her larvae there. And their larvae feed on decomposing material, very, very small amount. But it's just like how these things happen upon these in the first place to enable some to survive, to enable it to go down this evolutionary route, I just find remarkable.
Too right. I'm going to try and bring us back on track now, because now I've got one more question to ask before we go back outside the fly, and then you
Okay.
This funky looking thing here.
Yeah,
I mean,
lovely
it
ovaries.
license, it might not look like that, but what is this? The brown thing.
So the ovaries are amazing. I think what we've done is we've spent, I'm not immune to this. I spent far too much time looking at male genitalia. And really what we needed to be doing was looking at what's going on in the female. And what we've got with flies and a lot of other insects and animals and us is that you've got a cryptic female choice going on. So it's what's going on inside a female that's quite funky. And they've got, a lot of them have spermatheca, so they store sperm. And in reading it, my head is like, wow, this is amazing. I have a really bad imagination or a really good imagination, I don't know what you call it. And all I imagined is this room full of vats. And you've got nursery cells next to the sperm, and they stir the sperm to make sure it's still healthy. So I've got like little women like stirring, like, you know, like witches stirring cauldrons of this sperm, trying to keep it going. And then she decides which sperm she wants, and this is all chemical processes, whatever. She can eject sperm if she doesn't like it. And it's just fantastic what's going on there, the ability for them to do this. But the competition between the sperm and the egg and her, and she's trying to do that, and a lot of the sperm manipulates her, it manipulates her behavior. So then they've done this a lot with the Drosophila as well, and there's this, once she's had sex, the sperm is saying, right, you don't want any more. She's like, do I not want any more now? And it's like all of this going on inside these tiny little organisms. It's amazing.
Right. Okay, so I'm going to stop sharing it, and we're going to go out of the body. The bit I'm going to talk about.
Are we going to have an out-of-body experience?
I'm going to have an out-of-body experience. To be honest with you, as you were talking there about the sort of sperm, your fantasy world, I was seeing Charlie in the Chocolate Factory.
Ruined it now. Ruined that.
I know. I daren't mention it, so I won't.
You are. And guess what's going to come on TV at Christmas? Charlie and the Chocolate Factory. And every single kid would be like,
yeah, fly sperm.
Yeah.
Right. I'm just gonna, before, because next I'm going to talk about sort of does relate to this bit down here, on the female. So I'm going to stop sharing, like that. Now then, so much better than Skype. You can send the check in the post. So, ovipositors. This fascinates me, because you see these things flying around with these ovipositors, and all right, some of them look quite formidable. So you think, all right, yeah, I can see that. But they look, on some of them, so delicate, and yet they can pierce flesh, they can pierce wood, they can pierce. How does that happen? I mean, I know we talked about the exoskeleton, but that's fine. So it's got an exoskeleton, so it's can be quite hard. But where does the strength come from to be able to push the ovipositor?
No, this is what they're doing with the smart needles, and realizing that actually, if you slight pneumatic motion, you don't need to use as much strength as just pushing in one field motion. And I'm trying to find a nice ovipositor for you. Most of them are serrated.
Yeah.
So they're really good at slicing through other bits of body. I've just got a curly whirly penis.
That's not a sentence I thought I'd hear today, but anyway.
Curly whirly, you did. Come on, you did.
Yeah, I suppose I did.
And there's only one picture in the book. So the ovipositor is the egg-laying tube. Okay, so this is what we mean by that. So obviously it's only the females that have it. And the group of insects known for their ovipositors are the bees, because it's in the honeybees, it is the female, it's not the queen, that their egg-laying tube has been modified into a sting. Now, Diptera don't have a sting, but this looks quite funky and quite hardcore. And it's across all the females. You know, some of them really do look like they're wasps because they have this beautiful tapered abdomen ending with this nice ovipositor. Now, she lays her eggs into fruit. So a lot of them, it's quite nice and soft. But as you said, some of them lay them into trees. Some of them were my favorite groups. They're affectionately, their normal name is the bee grabbers or wasp grabbers. And she will basically use a tin opener, ovipositor, to slice through the thorax to lay her egg inside that, which is quite fun. Horrid phorids, affectionate name for them again, just because they're really difficult to identify. Some of them, again, they will slice through the thorax of an ant and the weakened part to lay in their eggs. This goes on to decapitate the ant. So amazing modifications going on with their ovipositor. So where there's a will, there's a way. She will find a little place to lay her eggs.
And you've half answered the question, right? Because, and it's a good half of answer, but you've also alluded to the other part of my answer, my question, which is still in my head. And that is, okay, so you've mentioned tin openers and sewing and all the rest of it. The mechanism that they must have in their abdomen, right? And I might be going too deep now. It's just that I have to overthink everything. Think about the motion that that fly has to go through in order to create a tin opener or sew a hole or whatever. The muscle, the combination of muscles there must be incredible.
I mean, this would have happened gradually over time. There would have been some sort of like mutation. It would be like, oh, that works. Or someone would have done like this. I mean, it's not like it was a conscious decision on their behalf to make these amazing structures. Mm-hmm. These structures work so they became amazing. And that's how evolution works. So yes, it would have been. The males, basically, some of them operate like pea shooters with their sperm. So it's like, I'm sure that's the noise.
That was... I can never see David Attenborough doing that when he does his life in the undergrowth or something. No. And this is how it works. That's perfect. And
so you know, she doesn't. Well, she does. She has to do that. A lot of there's some that the larvae themselves crawl out, so they don't have to worry about it. The larvae is the active stage of getting out there. But yeah, for it to be pump action into its environment is quite amazing. I describe basically the acrocerids, these little spider killing flies. This is a UK species. Look how tiny it is. It's a little hunchback.
All right, yeah, I can sort of see it.
Kind of see it. It's a little hunchback fly. She's quite adorable. She will lay thousands of eggs at one time. She's basically got a submachine gun as an ovipositor. She fires them out. She's exactly what I'd be as a mother. Absolutely terrible. And she's just trying to get as many out as possible because she's going on the strategy that if one survives, that's good. No care, just throws them. Great.
Do you know, we could go on for hours and hours and hours. If ever there was a phrase we've just still skimmed the surface, if ever that phrase was made for something, it's made for both your book and The Life of Flies, because you know, we have skimmed the surface. But you promised us a botfly.
Mixed diptera flashy. This is when I oh, not in this box. This has just got this is a massive hoverfly.
Oh wow.
Yeah, I know.
Where's that found?
Not here.
Oh right, yeah, it's useful.
Malaysia. Obviously everything gets a bit funky. Oh, I haven't got the... Oh, yes, I have. You can't really.
It's so this is a botfly. Is it a botfly you're showing us? Because it looks almost like a bee fly.
Just imagine a bumblebee. It basically looks like a bumblebee. It's absolutely adorable, and there's some beautiful images of the botfly. I'm not sure if I've got the botfly adults in here. That one is a reindeer botfly for Christmas.
Right, is that the one with the antlers?
No, its larvae live in the nostrils of a reindeer.
Oh, right, okay, so yeah, just so you
Why
know.
do you think Rudolph has a red nose? It's because of these.
That's why. And just so everybody knows that I'm not going bonkers, there are flies with antlers.
Oh, yeah.
Also flies with eyes on stalks.
Yep.
Go figure.
Yep.
But anyway, you got to, if you buy and read the book, and you'll understand. I'm flicking through the book because I want to leave you with a fantastic image at the end of this, which I love. It's absolutely amazing. And the description of it is brilliant. But we've got to leave it there because I honestly, I haven't a clue how long we've been talking.
Over an hour.
Over an hour, right. There's me thinking, I really need to get a grip on how long these interviews are. But I have to say that we could literally...
We've only done the insides. We should do another one for the outsides.
We will, definitely. I will interview you every week for 52 days. And I tell you what, we will still not cover it all. And it sounds like, look, you'd expect Erica to say that because Erica is a specialist and Erica has written 2 books. I am not. But these books are brilliant. And the things you learn about flies, it's endless. It's like you said about your partner. They're not only endless, they're also fascinating. So they're endless.
Yeah.
There's just so much to learn. And you think, all right, I'm a birder. That's where I started, but now I'm into all wildlife. I've been for about 30 years. But you think about birds and what can be said about birds. And I'm not dissing birds because I love birds. You know, birds, you could take up maybe, a few hours interviewing, but flies, it's a multiple of that. And you should go out and buy these books, read the books, study flies, amazing subject. So Erica, thanks for the book. Thanks for your time.
Thank you.
But you're about to say something.
No, I was just thinking about primatologists. There's an awful lot of them, you see. And I go into the field with a lot of primatologists. And I sat there once with a primatologist as they spent 3 hours watching a monkey eat. And I was like, okay. And then I took on my little walk round and I was like, now this fly. And I know I was looking at multiple species, but I'm sure still, if we properly started looking at individual species, just even, you don't have to go to a jungle for this. As I say, your garden is a jungle. Just go out there and start observing, and it is just brilliant. It fills me with absolute joy just watching all these creatures.
Thanks for your time. Absolutely. Thank you. Amazing interview. I will definitely be putting more interviews in the diary with you because I still want to investigate more. And we do want to talk about the outside. There's still more to talk about the inside. But I do want to talk about, in fact, I'll ask you a final question. Right. Okay. It's a big question. Answer it short, and then that will set us up for another interview. But,
okay.
So, you've already answered this in the last interview, but remind me, how many different species of fly have been identified in the world?
Oh, I don't know, between 170,000, 180,000, 160, a lot.
Right, okay. And that's fine. And they've basically, as we talked, when we talked about the brine fly, they've basically, they've taken sort of pretty much every niche of potential habitat, of way of life that
terrestrial
the Earth
niches
can possibly offer. So the Earth can only offer so much. So why do we have so many flies? Because you think of things and they evolve to sort of into a niche, but that's a lot of flies. So why do you think there's so many flies?
Oh, honey, that's not a lot. When we actually get around to describing them, there's going to be way many more species.
Exactly, yeah.
They've got flies, like nothing more than sex, and being nosy. And those two traits have enabled it to explore and diversify. So you can have rapid species turnover or rapid generation turnover and things like that. So you've got an amazing ability to adapt and adapt fast. You have the ability to fly into new habitats. You have great sensory perception, and you have a larval stage that is amazing because it's active. You think about bees, their larval stage don't do anything. But you've got an active larval stage that is consuming in its own right and living in a separate ecological niche in many times. So this has enabled these species to explore and diversify and to become nosy and do all sorts of amazing things because of their behavior and because of their morphology.
That was a good, short, succinct answer to what will be your fourth book. On that note,
yeah,
the books are brilliant. Thank you very much. Thanks for your time, and I hope to speak to you again in the not too distant future.
Thank you. That was great fun. Thank you.
Bye.
I'll go back to barcoding flies now.
Yeah. Nothing I can do today is going to be more exciting than that. Take care. Bye.
Bye-bye.